Welcome to What's New in MPMI, which is the virtual seminar series of the MPMI Journal. And it's a pleasure to have you all here today. Before we get to our main event here, which is a really fascinating talk by Li-Jun and graduate student Houlin, we want to just update you on a few things that are coming up. And I'm co-hosting today with Tessa Birch-Smith, who is Associate Editor-in-Chief of MPMI. And Tessa, I'm going to turn it over to you to tell us a little bit about this upcoming focus issue that we are currently gathering papers for.
Good morning, all. Thank you, Jean. And I'm going to quickly share my screen. So MPMI is pleased to announce its 2023 focus issue on the plant endomembrane system in molecular plant microbe interactions. We have a deadline of June 30th, 2022 for submissions, and we are pleased with to have an excellent slate of focus issue editors. They are Li-Jun, Dr. Yoon-Sun Park, Aiming Wang, and Dong Wang. So we encourage all of you to submit your research articles in this area, and you have the benefit of being part of a dynamic focus issue that will get eyes on your work. So please consider us if you have any manuscripts in this area. Should you need any special considerations, please feel free to contact myself or any of the focus issue editors. And I'd like to make a second announcement, and this is a reminder or an announcement in case you're not aware of it, that in 2022, the International Society of MPMI is very pleased to host an early career showcase. And this is for senior graduate students and postdocs. We will have two events. One is from June 8th to 9th, and the other will be in fall in September 20th to 21st. And in each of these sessions, we're hoping to hear from 8 early career presenters in the field of MPMI. So this requires nomination before selection as a presenter. So you can please nominate yourself or nominate a colleague who you think will do an excellent job presenting your work. So this MPMI is very excited about this because it's a chance to profile and give encouragement and exposure to the fantastic work our young members are going to do. And we get a small taste of that today with Houlin's presentation this morning. So thank you all very much.
Thank you, Tessa, for sharing all of that. And I believe we have the links in the chat, so you can go there if you want to read the information in more detail. And that reminds me that MPMI is offering a special discount for the first half of this year to papers that are submitted with a student first author. There will be an $800 reduction in the cost of your manuscript should it be accepted for publication. And this is because it's part of MPMI's goal to promote the careers of our young colleagues. So I hope that you will consider that. And Greg has posted information about the student discount into the chat. And I want to draw your attention to our next What's New in MPMI seminar, which is coming up on April 4th. So a month from now, Preeta Ganguly from Moichi Dasgupta's lab in India will be presenting on her recent MPMI paper, and she is also a graduate student. So we have are trying to promote the careers of young researchers in our What's New. Finally, before I turn it over, we start the actual seminar, I want to ask for volunteers for today's seminar by Li-Jun Ma and Houlin, volunteers to help us edit the transcript that arises from this, that Zoom creates. And that's to increase inclusion for people who are hard of hearing or for whom English is not a first language. These transcripts are really invaluable. And as you know, Zoom only does so well with scientific language. So please, if you are interested in doing this, please, you can contact us in the chat, or you can also email me or Tessa Birch-Smith, and we would be very grateful for you volunteering your time to help improve inclusion. which is a big goal of our seminar. Okay, so very glad to see everybody joining in from all over the world today for a very special seminar. I was tremendously excited about this paper when we had it for MPMI, and it's a really interesting topic, taking us thinking about these questions of some of the big questions, the big top 10 questions of how can we, what's the difference between pathogens and very, very closely related non-pathogenic organisms? How does that work? And takes us into the world of the pangenome, exciting new area to be thinking about. So I am going to turn it over now to Li-Jun Ma, professor at UMass Amherst, and also one of our senior editors at MPMI, who focuses on articles around that focus on genomes and genome announcements. And also Houlin Yu, graduate student who was one of the lead authors on this paper. So thank you, and I'm turning it over to Li-Jun.
Thank you so much for this great opportunity. I think the whole interaction has been so rewarding. But for the time matter, I'm just going to go ahead, start sharing my screen. So this is a paper published, oh, take a while. This is a paper published in MPMI early this year, and the paper was led by two talented junior scientists. Li Guo was a postdoc when the project started, and Houlin is here, did a lot of data analysis process. Why I'm here, so Houlin is here, can do that. So I decided to use a little bit time to propel the audience why we entered the space and what was the thinking behind us. And actually, this is so closely connected with this top 10 questions. I love this paper, Dr. Harris, and leading the process, put it together. And I wanted to invite us to think about the excitements as well as the challenges we are facing today in addressing these questions, or maybe some other interesting questions you have. And then, so therefore, what make me thinking, what is special about today? This weekend, I had a great time meeting an old friend. We was reflected what is about yesterday, right? So in order to see what's special about today, let's compare something happened in yesterday. So a few things about yesterday we reflected when we were graduate student, what happened in there. So yeah, we have to type everything. And so in there, so you have to be careful with words because you went wrong and you have to retype again. And we have to go to library and write the detailed notes, flipping through books and old journals. Doesn't like today, you just go to internet, search the term, the paper will come to you. So it's very painstaking process. But worse than that is collecting our data. So I actually read many jails like that to read what is A, T, C, G. Luckily, when you graduate, you sequenced a few important genes you can publish, right? And I heard plenty of them. If you get a PhD, if you clone a gene, maybe that's good enough. So in summary, maybe you will agree with me. It's very slow. For today's graduate student, maybe you will add a painfully slow behind that. So then what is special about today, right? That's just not talking about the internet enable us to the computer stuff. Let's just thinking about data processing. So compared to the one I have to read this gel, you'll probably get the data just pop into the inbox like that. And you will basically have as much data as you wanted or you needed. And you are swimming in the ocean of data. It's very often your PI will come to you to say, I need one more piece of data. Or maybe sometimes you said, I wish I have one more piece of data. What does that mean? Not necessarily you fall asleep in this race, but it could make you lost or confused how to get the meaning out of this ocean of data. So today I wanted to emphasize comparative approaches to help reduce complexity And this paper published in NPMI is one way we kind of exercise this concept, reduce the complexity through comparative approach. And so that come back to the system we work on in the lab. So my lab focusing on Fusarium oxysporum is a cross-kingdom pathogen. It's Ascomyces fungus. And interestingly, the fungus has a unique feature is they reproduce asexually using 3 different spores, but we still not able to reproduce it sexually. And as a species complex, Fusarium oxysporum causing disease called Fusarium vascular wilts. So here are just a few examples, and they could infect over 100 different plant species. But interestingly, that they are maintain very strong host specificity. What that means is, if we have a pathogen that infecting banana, they will not infecting your tomato, vice versa. So because this case, so then we give a special term called formal special alias. So currently there are more than 120 different formal special alias existed in the literature. And the reason we call it cross-kingdom pathogen, they also infecting humans. That's not the focus of today's talk. In 2010, we using another comparative genomics approach, we sequenced one of the Fusarium oxysporum genomes. There we identify something we called accessory chromosomes. So here is it's a busy figure. What we show is Fusarium oxysporum genomes are highlighted as black bars, and the color bars are sister chromosomes. So if you see a color bar pinned to the chromosome, which means it's highly conserved in the sister species. And here highlight 4 accessory chromosomes. They do not have homologous chromosomes in the sister species, but they are carry very known factors. And then when we go back to sequenced many different Fusarium oxysporum genomes, we find every one of them carry accessory chromosomes. Related with NPMI topics, I have been always wanting to understand molecular mechanism underlying this Fusarium and the plant interactions. But look at here, where do I get started, right? Which host, or do I study every single host? Everyone is interesting. So where to understand, where to start to understand this interaction? So again, so I'm searching for the comparative angle, just looking at NPMI, the top 10 questions. The number 1 is beneficial versus pathogenic. So we were thinking, okay, thinking about that, this pathogen is host specific. So therefore, most strains are not pathogenic to the non-host, right? And plant actually wins most of the time. If I thinking that way, so then host does not matter that much, and it's important to forming this pathogenic and endophytic interaction system. And this is started by postdoc Li-Jun and the graduate student Catherine. And so we try to using the model plant Arabidopsis, and we have a biocontrol strain F47, which does not causing disease, while the pathogenic strain were infecting the plant and kill the Arabidopsis effectively. And Catherine did dry weight measurement, actually demonstrate that the biocontrol strain make the plant a little stronger, while the pathogenic strain killed it. Does not grow very well. So now we're going to, because we are the genomics lab, we empower the power of the sequencing. We wanted to use metagenomics. What that means is we want to look into all genes, both fungal genes and the plant genes, and involved in this interaction. And we want to see, are there the same genes or different genes from the same host, Arabidopsis? Do they react differently for these two different interactions. We also wanted to understand the contribution of the fungal accessory chromosomes. With that, I'm passing on to Houlin.
Thank you, Li-Jun. So here we start to ask what are the molecular mechanisms underpinning the different plant phenotypic responses to endophytic and pathogenic Fusarium or C-sperm, which genomes that share the core but can be differentiated by the accessory. We took a Metatranscriptomics approach, conduct RNA-seq of Arabidopsis roots inoculated by two strains, as shown here, Endophytic 47 and Pathogenic 50 and 76. And we collected the roots at 12 hours, 24 hours, 48 hours, and 96 hours post-inoculation. At 12 hours, we also inoculated Arabidopsis roots with water as a mock control. We use this data to disentangle the differences and the similarities between endophytic and pathogenic interactions. Li-Jun, please go next. We first used all the differentially expressed genes to generate a global comparison between different conditions. As you see here, the red circled, when we compare the pathogenic and endophytic interactions at the same time point, the transcriptome was very similar, as indicated by the relatively high correlation score. Li-Jun, please go next. So we next compared endophytic and pathogenic interactions at different time points individually to the mock. At squared in red, you can see that we have observed a low correlation when we compared both fungal treatment at 12 hours with the mock. And as the time goes on, you can see the correlation become higher. This actually indicates the plant have a very strong response to both types of fungal partner, no matter it's a pathogen or endophyte. And then the transcriptome somewhat go back to a normal as time goes on, comparing to a mock. And this is all within like a short 4-day time course. Li-Jun, please go next. To probe the genes and the expression patterns related to plant defense, we first conduct a k-means clustering and try to separate differentially expressed genes into 24 clusters. And here we also perform a goal enrichment assay and have identified, has been indicated by the circle, there are 4 clusters. As they are enriched in goal terms related to the defense and the stimulus response, and also at the same time they are lacking the goal terms related to the development. Li-Jun, please go next. We then look at the gene expression pattern of these defense related clusters. In each plot, the middle is the expression of the mock at 12 hours. And from the middle to the left, the figure illustrates the expression endophyte treatment from 12 hours to 96 hours post-inoculation, as indicated by the directed arrow. And to the right, the figure illustrates the expression under pathogenic treatment from 12 hours to 96 hours. As you can see, the genes were upregulated within cluster 7 and cluster 15 on top at 12 hours, and then gradually turned back to a mock comparable values. And the genes were downregulated within cluster 16 and cluster 21 at 12 hours, and then gradually turn back to mock comparable values. Although there you can see the difference is in cluster 7, cluster the 21 and the 16, there are differences between the 2 different treatments. From here, we also see that the cluster 15 illustrate a very similar expression pattern between 2 treatments through the time course. And it worthwhile to know that this is the largest cluster with the largest number of genes within it, which is also consistent with the early discussion I showed that plants respond very similarly to the endophyte and the pathogen when you look at the same time points. Li-Jun, next, please. We then zoom in to the goal enrichment to these 4 clusters. Not surprisingly, you can see that cluster 16 and 21 are more similar to each other than comparing to cluster 7 and cluster 15. And 7 and 15 also share some goal terms, such as catabolism, cellular homeostasis, and which genes encoding proteins localized to the plastids. And also cluster 16 and 21, which are similar, they are undergo a strong suppression by Endophytic, shared many terms related to signal transduction, which are enrichment of genes encoding proteins localized to the plasma memory. Next, please. Since the largest cluster with shared expression patterns can represent the common immune responses by Underfeed and pathogen, we therefore carefully inspected the gene list and identified genes as indicated here related to the receptor like kinases, nucleotide binding leucine repeat proteins, also known as R genes, resistant genes, and also the proteins with chloroplast functions. Since our mock at 12 hours underwent the same experimental procedure as 12 hours fungal treatment, the observation of the genes related to the chloroplast functions shouldn't be an artifact. Next, please. When we carefully examine the list within cluster 16 and 21, there are many well-characterized genes, as shown here, involved in the danger sensing and signaling from the signal perception, cellular signal transduction by kinases and R genes, and salicylic acid biosynthesis and signaling. So this further demonstrates the power of Endophytic in suppressing the plant immunity. Li-Jun, next, please. Since 12 hours is the most distinct time point from both global correlation of differentially expressed genes on the left and also the expression plot of 4 immunity clusters on the right. So we decided to using this 12 hours expression of mock, Endophytic, and pathogen treatment further to compute what genes are specially or more extremely upregulated or downregulated by Endophytic or pathogen. Li-Jun, next, please. So here, when we ask what are genes are significantly downregulated by Endophytic comparing to both pathogen and also mock, we computed the gene list and then characterized the goal enrichment showing as a network here. So the figure, each node represents a single goal term, and the same color indicate the goal terms actually largely share the functions. As shown here, there is an immunity module by the goal terms related to JAC response, immunity, defense, and etc., which is not surprising, as I already showed that Endophytic is good at suppressing plant immunity. Li-Jun, next, please. Then what is especially or more extremely downregulated by the pathogen? We here identified a module related to cell division, represented by a couple goal terms related to cell cycle, cell wall biosynthesis, which is consistent with the phenotype Li-Jun showed early that Arabidopsis actually the growth was perturbed by pathogenic strain. Li-Jun, next, please. As shown here, there is a module related to the plant defense, which has been more extremely upregulated by the pathogen. And when we look at the goal terms, the term related to the for example, the toxin metabolism. So therefore, the pathogen downregulate the plant growth and activate some part of the defense, as you can see from these and the last slides. Li-Jun, next, please. And finally, here is showing a strong nitrogen metabolism model, which was very interesting and is only associated with the 47 of the endophytic strain upregulation. I think we think this may explain the growth promotion phenotype that Li-Jun shared at the beginning. Next, please, Li-Jun. So after looking at the plant transcriptomic response, we then want to get like some idea why the plants respond differently. So assuming if we have like two strains with the same genome, there won't be any differences. So therefore, the differences we observe here so largely can be correlated to the differences of the genomes of the two strains. And we know the two strains of the Fusarium oxysperm, they share the co-genome. Therefore, like we focus our analysis on the accessory genome of these two strains. So here, as you can see, we plotted the expression of genes that will not only show the protein domain enrichment in accessory genome. So by enrichment, I mean there is a significantly more numbers of those type of domain containing genes in the accessory region than if the domain is randomly assigned to the accessory genome. So not only the enrichment, but also we have a criteria to filter out the genes of induction during the plant colonization. So interestingly, you can see Endophytic 47, it turns up the genes mostly related to the signaling, such as the G protein signaling and the transcriptional regulation. And meanwhile, you can see the pathogen 5176 turn up we can summarize as the degraders as the terms related to the peptides and the proteases and as well as the effector proteins. So sort of facilitate the fungal partner to infect the plants. And there are also, interestingly, we found the domains such as the PARPs and the WGRs. They are all related to the DNA repair, which are also enriched in the pathogenic accessory genome and are regulated during plant colonization. So the differences showing up here likely contribute to the different response to the pathogen comparing to the Underfeed. Then I will pass the stage to Li-Jun to wrap it up.
Okay, thank you, Houlin. Oh, yeah. Thank you, Houlin. That was nice. So I just kind of catch up. Houlin mentioned that this pathogenic 5176 genome, and then this is 47 endophytic genome. And when you see a line here, means the two genomes are almost identical. So highlights like 47 has its own accessory chromosome, about 4 MAC base, and the pathogenic genome has about 20 MAC base. It's much larger. So I thinking this coming back to say, wow, majority of these two genomes are identical, right? And Houlin had been showing you that plant does know that they have two Fusarium oxysperm infecting them. So here's the summary slides for this paper. And the take home is that 80% of Arabidopsis genes just behave the same. Doesn't matter it's a pathogen or endophyte. So if we're thinking, Houlin have been done beautiful job streamline the result, but if you're thinking how many data points he has to deal with, it's amazing. But since it goes through this process, we already understand 80% of the genes, they are important. If they're not important, they wouldn't be there. But they are not involved the different interactions between this pathogen and this endophyte. So therefore, we can focus on the 20% of the genes. By mining the 20% genes, we know that the endophytic interaction can attenuate host immunity, but promote the plant nitrogen assimilation. In contrast, the pathogen activate plant stress response, but suppress plant growth and development. So I think in this powerful comparative angle enabled us to draw knowledge, correlate the genotype and the phenotype. At this level, of course, lab is continue going on. There are many questions to follow. So here's the lab members. Li right now is a professor at Beijing University, and 2 new postdocs joined it in. So Shira is doing DNA repair, and Domingo doing more Fusarium, and plant interactions. So we had a lovely summer retreat. And of course, most of the work presented here are sponsored by National Science Academy, and lab have been fortunate funded by other funders. And I wouldn't be here, Houlin wouldn't be able to join me without all wonderful collaborators. So I just list here without name everyone one by one. So that's it for us.
Thank you, Li-Jun, and Houlin for a really fascinating talk. I just love this idea of the accessory chromosomes in these two very similar isolates directing all of this. Why don't you stop sharing your screen, and we will, oh, there we are. And we'll start to take questions from the audience. You can type into the Q&A with your questions, and we will be using the chat just for other kinds of correspondence. And I also want to say thank you. We've already received one volunteer to edit the transcript, so that's wonderful. Thanks. While we're waiting for people to type things in, I'm going to start with a question for Houlin. When you went into this, did you have as a hypothesis that you might be finding accessory chromosomes, or was this something that you discovered as you tried aligning the genomes?
So yeah, that's a really good question. So I think before I enter, because we know from a previous study, like the Fusarium system, they share the co-genome. So therefore, when we enter, we try to focus on the accessory genome because they are largely different. So we have some idea, but we didn't have like a clear idea what we can find. And after we got the data, like the pathogenic strain more enriched in the degraders and like the DNA repair, and the endophytic strain is more signal. That was like a amazing result we didn't expect early. Yeah.
Yes.
Okay, so we have our first question in coming into the Q&A, and it's from Amit Kashwani, and he asked, which types of genes are highly expressed during pathogenesis? So I think you may be looking at slightly later time points in the infection. Although your work focused on 12 hours, I think he's asking about later on with the pathogenic strain. So maybe about 72 or 96 hours.
Do you want to take it, Houlin?
Oh yeah, I can take it. So the first thing is like I think our study focusing on the very early response, like from the 12 hours to 96 hours. And actually at 96 hours, when we look at the plant, they are not although we see the downregulation of the plant growth and upregulation of the metabolism, toxin metabolism, like there is not a huge overall differences. So and also we found from this comparison, like we found that there is a actually strong induction at 12 hours, and then those genes downregulate, and this part of gene related to the some defined signal, and as I show, the chloroplast related to the defense was the most interesting thing, and we didn't quite look at at 96 hours. Uniquely at 96 hours, but we think likely those toxin metabolism is still going on at 96, and also they're like if we expand the time course to like a much later time points, like 6 or 7 days. Maybe after like when you see the phenotype, there may be another set of gene has been like induced. So for this analysis, we try to focus on the early time points, and it's correlated with the chloroplast, all the some signaling genes.
So yeah, I can follow up a little bit on that. So this study, we really using the early time. So 4 days is maximum we reached. Most study will go later because the plant died when that time. And the reason we focus on this time frame, we really wanted to know when did the plant know that something is there? And then when they realize something's there, what's their immediately response toward that? So actually, if you thinking, that's where the complexity is, right? Plant not just deal with the fungus, they are still keep growing. And the fungus not only try to kill plants, they try to grow as well. So they are complete lives there. So all the genes, many genes are active. So therefore not involved in the difference, has enabled us to dissect the difference. To answer this particular question, we do know that if we're looking at 12 hours, many of the characterized effectors are not have the highest expression. By 96 hours, you see some effectors start to peep up. But some other labs have been looking for 7 days or 10 days. The effectors probably even higher expression. So we are trying to understand other effectors which haven't really fully focused in the literature. They are highly induced. So what they are doing. So I don't think we understand everything involved in this interaction, but it's more dissecting the effectors in different groups. So right now we have an active effector study, try to characterize them, and based on their structure, expression, and everything we can put, and try to understand more of the genes involved.
Yeah.
So fascinating. I love the idea of aiming so early so that you can actually capture what happens from the first moments where the plant says, oh, there's a fungus, to where it says, oh, what kind? And the fact that they share so many similarities, so interesting. And I think those effectors are going to prove really, really valuable. So we have a question coming in from Jesse Kaliku, who asks, is it that the endophytic strain evolved from the pathogenic strain or vice versa?
Only you want to try?
Actually I'm not expert on this. Li-Jun, you can give some idea.
So yes, so that's actually a fascinating question. In 2010, once we sequence the genome, we see, wow, extra chromosome for the pathogen. So then immediately thought is that then non-pathogen shouldn't have anything. So actually discover this 4 MAC base accessory chromosome for this endophytic strain was a surprise to start. But it's true. So when the gene asks, what's your hypothesis, right? For some of the genomic project, it's really data driven. So I think first of all, see that was a surprise. And now we are dive into, so now there are hundreds of Fusarium oxysporum genomes existing in the database, some from us, some from other labs. It's all around the world. And then, so we start to say, where you focus, right? So this is a great question. That's 47 need to have this actual chromosome in order for it to be a good endophyte. So we need to look at more endophytic strains. I think in the group in the Netherlands, they did a lot of endophytic strain sequencing. And we feel like, who is more important, right? So we start to dissect the kinases, transcription factors, effectors, one step at a time. They probably all important. So then where, who get what? One experiment was done was using pathogenic strain, the accessory chromosome from pathogen force it to migrate into the non-pathogen. You demonstrate if that happens, you're going to be disease. You're going to cause a disease in that specific host. But so in order to test the hypothesis, this actual chromosome from 47, it's the reason make it endophyte. You have to remove the accessory chromosome from the pathogen and transfer this endophytic chromosome into that one. So we haven't set up to do that. And I guess every time people have to ask me, where do all those accessory chromosomes come from? I would say, if I can answer that before my retirement, I would be very happy. But we don't fully understand. So that's true.
Yeah, it's a complex system, right? So
Absolutely.
yeah, so you can have complex outcomes. Okay, so our next question in the chat comes from Ankita Alexander. And Ankita makes an observation saying that in their study with plant growth promoting bacteria, I think it is, they also see high expression of photosynthesis related genes. So then the question is, do you think that the plant uses a common pathway involving photosynthesis for both pathogenic and beneficial interactions? And both of you are nodding furiously. So Houlin, you want to take a stab at it?
Oh, sure. Yeah, it's a very interesting question. So when we analyze the data, we saw the very strong signal of the photosynthesis genes has been upregulated, no matter it's pathogen or endophyte. So we do believe there is a very strong, you can call a general immunity or something like that, between the plant and the fungal partners there, involved in the defense or sort of the recognition. So when we look at the genes, we find, although it's termed photosynthesis, but all the genes actually target the plastids. So if you consider the root, so they don't in under soil, they don't undergo photosynthesis. But as the research going into plastid immunity, we do believe the plastid serve as a immune hub or something like that to as a recognizer or responder to the fungal partner. And there is very strong signal, also very interesting. So Li-Jun, you want to add something more?
I think you did a good job.
Thank you.
It's an interesting point that I always try to bring out in my plant physiology classes, that all cells have plastids. It's just only in some that they're chloroplasts. And of course, you brought up the whole idea of nitrogen assimilation, and a lot of nitrogen assimilation happens in the plastid. So it's very interesting to think about that former endosymbiont as being something that is being manipulated by this current endophyte. Interesting question. Okay, so let's see, we have a question from Ashajyoti Mushineni, who asks, do the plants respond differently to fungal and bacterial endophytes? And really asking, is the phenotype that we perceive, will it be similar or will it be different?
One of you want to try?
Yeah. So it's very interesting question, endophytic interaction. So we haven't including endophyte, the bacterial endophyte as a comparison, but we do do some reading, we find in the bacterial endophyte interaction or the Arabidopsis actually respond we can also saw in those papers they set up a regulation of the nitrogen and also the phosphate. So there are some interesting papers in phosphate as well. So what we didn't discuss here is we also observe the phosphate induction by the endophyte as well. Although it's not that strong as the nitrogen, but certainly we do believe there are some similarity in terms of the endophyte interaction. Yeah.
I just want to add on to Houlin's point is that when we think about bacterial versus fungi, plant recognize them differently. So you will see the receptor start from beginning, who is interacting with me will be different. So then the downstream pathways activated by the initial recognition could be different, but doesn't matter. It's a fungal plane action, all bacterial plane actions. The outsider usually was sending a bunch of actors into the plant to manipulate the system. So when that happens, intersect all the time. So like Tessa would say, some of the virals also attacking the chloroplast of plastid. And so I thinking that effectors, that's why the effector biology is fascinating. So all those microbes will deploy tons of proteins into the host. And then they find their target to manipulate. And many of the targets are the same. Doesn't matter if it's from bacteria or from the fungi. But I think the initial recognitions are different, and therefore the interaction will have differences. That's why MPMI is a fascinating subject.
Indeed, and you have done a fascinating study. So I just want to say we have someone who has raised their hand. If you don't mind, if you have a question, please put it in the Q&A and we'd be happy to address it through that channel. So an attendee asked, can you please explain why the pathogenic Fusarium is very specific to the host? On the other hand, is the beneficial Fusarium also host specific or not? Where does this host specificity come from, and is it universal among the Fusarium?
You want to try, Houlin?
Oh yes, sorry. It's like one of the fascinating question, like host specificity or the specific pathogenicity. Right? So the first question asking like basically what determine the host specificity or our strains? Yeah, so our strain is a host specific to the Arabidopsis. So when you inoculate to other crops like tomato, it doesn't causing the disease. So from the study, like we know the host specificity can be correlated with the accessory chromosomes. So some component in the accessory chromosomes control this. So we know something like the effectors play a role. So if you knock the effector out, you certainly lose the pathogenicity to that host. And also then the question like from here we still have is like some component that effector, if you knock out, is for the host like pathogenicity. So then what determine the host specific pathogenicity, right? So I think it's still like a big question there. Although like if we do the comparative genomics, we can know some differences like in the effector, we call effector repertoire in the accessory chromosome. So basically the same host, if you cluster the effector together, we can find the like they have the same effector patterns and something else. So that can be determined the host specific pathogenicity. So and then the second question asking like the Underfeed. So is the Underfeed to the Arabidopsis also Underfeed to other host? So right now we also did some study like we inoculate the Underfeed to the other plant like tomato, and from the microscopy we can also see like Underfeed colonize very similarly to the tomato or comparing to the Arabidopsis. So we do think like there the Underfeed is an Underfeed to like not only to Arabidopsis but also to other plants. You want to add? Yeah.
Yeah, I think that's the key, fantastic question, which is the endophyte does not have host specificity. So it can go, so far. So this one, we are lucky. So one of our co-authors of this paper was leading on this FO47 study. This one was isolated as a by-control for many different plant hosts. So if you go through the literature, people have been used this one to control pepper disease, control tomato disease. So it doesn't matter. So like we didn't know they will be promote Arabidopsis growth. It did just fine. But so how could, right? So I think this is a fantastic question. Why the pathogen has a whole specificity, but this non-pathogen doesn't have to, right? So that's fantastic way to continue explore. So, but I think the difference is strong, it's significant. And we actually have RNA-seq for 47 into tomato, and Houlin is looking at that. Should we asking the question, the same genes from 47 are activated, the same pathway in tomato are activated, but I don't know. Houlin is looking into that. Maybe next year we publish in MPMI with a follow-up.
That would be
That
very
would be
nice.
lovely.
We have to convince Houlin that's a good project.
It's a very important question. So Houlin,
I
you should consider it.
Very important question.
Yeah, very interesting. And it's a great question, thinking about host specificity with the endophytes. Very interesting to see what broad spectrum it has. Okay, we have a question here from Amit Kesharwani, who is working on Xanthomonas and studying plant pathogen interactive genes during pathogenesis in the host, and is wondering if there is any software you could suggest that provides in silico analysis to characterize them. And then he has a follow-up here, host R genes that interact with pathogen avirulence genes. Any suggestions on?
Good at this. Go ahead, Houlin.
So the first question, my understanding is to ask like any software to like characterize the genes related to the pathogenesis.
Yeah, start with RNA-seq analysis, I guess.
Yeah. You can start with RNA-seq analysis, and I'm not sure a specific software, but it's a complicated processes. Like you need some like tools and coding, and but we like if you are interested, you can reach out to our lab or reach out to me in particular. We can discuss more like how to start like from beginning. So it won't be like a one tour solving like the whole process. Yeah.
Thank you, Houlin. And that actually reminds me to tell people that, of course, this one reason we have this is it's a great way for the audience to interact directly in real time with the authors. But remember that each of our papers has the email link for the corresponding author. And that's a great way to contact them to ask your detailed or specific questions about approaches, et cetera. And they welcome these. So. And this is my other little plug, which is to say that graduate students who are watching or undergraduates, I think what Houlin is saying is wonderful advice, which is you really have to develop the skills to be able to use these bioinformatic tools, which means learning how to do some coding. And I think that should be in the curriculum of every graduate student out there, no matter what your topic is. We're only going to have more data. As you saw from the beginning of Li-Jun Ma's presentation, if you think back over the course of her and my career, how much data explosion there has been, it's really exploding. Next year, there will be a huge amount more, and so on and so on. And really, anything that gives you the skills to handle this enormity of data is really going to help. Okay, I'll stop with my little plug there and turn it over to Tessa for the next question.
Okay, so in the interest of giving everybody time to ask a question, I'll skip ahead to a question from Tim Friesen, who asked whether you know any specific effectors that are released by the endophyte. It's going back to our discussion about specificity. So does the endophyte produce any specific effectors, even though it has a broader range?
So actually, Houlin, I'm taking this one because I'm so warm with this topic. Because that's exactly the question fascinating to me. So we looked for the compared to effectors because the pathogen definitely have way more effectors compared to the endophytes. But I tell you, the endophyte do have the specific effectors. One of them very highly expressed at 12 hours, targeting chloroplast. So yes, but But do we know everything about it? No, I need a next grant to do that. But yes, I was amazed to find that candidate. So we try to clone that one, try to ask undergraduate students to study that one, and try to form in collaborations with the chloroplast scientists to really dive into that. But answer is yes.
Exciting. Waiting for the follow-up. Okay, Jean.
Absolutely. I am going to jump down to our last question, but we may come back if there's time to some of the other ones. So an anonymous attendee says, I wonder if you tried to analyze the transcripts corresponding to the fungus in this time course experiment.
Yes, we did too. Okay, Houlin didn't do a lot on the fungal side, is that right? So I think the postdoc did more on the fungal side. But yes, we do have the fungal data. Actually, Houlin's picture, the slides showing the expression of accessory genes are plotted over the time course. But I think it's the same. That project as itself probably will be taking a few months. You are interacting the whole system. And we have been doing the dissection, looking for the transcription factor expression differently, the effectors expression differently. We don't have as a beautiful story as this published one from the fungal side. But yes, there are stories to come.
Okay.
Okay. Do you want to
we have-
ask the next one?
One last question. And this is one that I second this question. So here it is. If the endophytic strain attenuates host immunity, could the level of attenuation make the plant more susceptible to other pathogens, including those that it was previously resistant to? And I was thinking about this, what if you did a mixed infection of the endophytic and the pathogenic strain? How would the plant respond? What would the outcome of that be? Which is more or less the condition in nature where you have multiple interactions happening at any time point.
Tony, you want to try? There are some literature data. We also have some data.
Yeah, it's a very fascinating question. So the point like the attenuate the immunity. So it's a immunity. So I would say the immunity can be different kinds of immunity, right? So the immunity we are discussing here is actually the people have been studying a lot, like MPMI, the field, like the immunity related to the sensing, signaling, and the downstream like a response. So when the 47 comes, actually, if you consider like another kinds of immunity, which is the cell wall strengthening, like the physical immunity, actually the 47 can make the plant stronger in that front. Like we do observe that like when we inoculate the 47 to the plant, not the Underfeed, actually can induce the like many the plant the cell wall components the deposition in the plant. So that actually is attenuated immunity as like a signaling part. However, it's upregulate the immunity as the barrier, like a physical barrier. So that part actually we believe they actually have to defend other pathogens. So when the people like we didn't do this, but the other researcher, when they inoculate the 47, the Underfeed, together with the pathogen, the disease will be suppressed largely, like very strikingly, comparing to the inoculation of the pathogen. So and we believe like the physical barrier immunity actually play a role there. So yeah, that's my answer.
Yeah, so I think just add a couple of sentences there is that 47 have been used by control. But how did it really actively helping plant to defend themselves it's more a little bit open question. So one study done very nicely is infecting the plant with oomycete and then putting 47 there. There they show clear demonstration that 47 can kill the oomycete pathogen. So that provides a control. And in our lab, we also wanted to see whether 47 can suppress the disease. Really is, how do you do that? So I thinking I had one master student did a little. So if you inoculate the plant with 47 before you inoculate the pathogen, you see protection. If you mix them 1-to-1 ratio, you see some protection, but not immune. So I thinking it's so many questions to follow. We didn't do the transcriptomics for the mixed population. So that's also unknown.
That is a fascinating question. And that really brings us back to our top 10 question number 1. And if people haven't seen it, maybe we can paste that into the chat so you can follow the directions to there. But our top 10 question was, how does the plant simultaneously interact with beneficial and pathogenic microorganisms? And that's still something that is so completely unknown. And I love the way that if we circle back to the beginning of this seminar, talking about the idea of using these comparative approaches to reduce complexity, I think is an incredible way to do this. So Greg has pasted into the chat the link on the top 10. So many of the questions, including this whole, what came up last, which is this idea of how does it all happen when things are out in the field? And that's important both from an agricultural perspective, but just in general, what are all the different factors that modulate this? And I think we are just scratching the surface on that endophyte, and that is going to be a long future of fascinating experiments. Hopefully we'll get the next one in MPMI. So we will stop there and thank everybody for joining us. It's been another tremendous seminar. Thank you, Li-Jun and Houlin for agreeing to speak. And of course to Tessa Birch-Smith, our Associate Editor-in-Chief. And if you have any questions about any of the announcements we raised, just email us, and questions on the wonderful seminar, you can contact Li-Jun Ma through the email link in her paper. Okay, well, thanks everyone. And we will see you in a month.
Thank you all. Great presentation. Thank you.
Thank you.